So I should say, I didn't really prepare anything major because Tony emailed me several days ago and said, don't worry, I have it covered. You don't have to do an introduction, blah, 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 blah. And of course, yesterday, said, I don't really prepare anything. I've got 4 slides. So it's not my fault. So I'm not going to give a general introduction on anything. I'm not going to give a general introduction on Evo Devo, and I hope these guys covered it actually quite well. I think I'll just add, after listening to what they talked about, is I think we all know that people say, oh, I know all about evolution. It's about natural selection. It's much, much more than natural selection. And it's more than just genes. And I think Tony pointed out that we're enamored with genes, and we want to make large inferences about genes and how genes control complex behaviors in the gene for language, and we have a gene for love, and we have a gene for all these sorts of things that humans engage in, and it's not that simple. And I hope as the week progresses, you'll appreciate that. So welcome to the BCBT. And before I go any further, I would like us all to say thank you very much to Anna and Miara, who had to put up with all of our shit, including the speakers, the students, people changing their schedules, and she's done a really, really awesome job. So thanks so much. Okay, so to understand brain evolution and development and the behaviors that emerge within a lifetime and across lifetimes, we can't study the brain in isolation. And that's something I've learned over the years. As a graduate student in John Caz's laboratory, I looked at brains. I listened to how neurons responded to different types of sensory stimulation. We looked at anatomical connections. And there's a tendency in science to focus on your own level of organization, whether it's molecular, genetics, systems. In order to understand these complex processes and how complex behaviors emerge, it's not enough to just look at one level of organization. We have to take a multidisciplinary approach and study multiple levels of organization, from genes to brains to individuals to societies. And that's becoming more and more apparent if we want to understand these, how complex processes emerge. So this is just a little introduction I put together some time ago. So we have our brain, but we have to appreciate that it is embedded within a body, and that the body, different bodies have different morphologies and different sensory specializations. And that brains act not only as my brain does not act alone. I'm not a human being in a vacuum, but I'm embedded in a society where more complex behaviors emerge, especially at sports events, where groups of people can do things that individuals can't do, and that we also live on a planet of not just conspecifics, but heterospecifics and other societies of animals in an environment that can be warm or cold, but we are all constrained by the physical or the laws of physics that operate on our planet. So those are very large constraints. And all brains have early experiences, such as language, culture, and social interactions. And we have decided, as a larger society, how groups of brains are going to be formed. We've decided this through our education systems, our system of social interactions, what is considered appropriate behavior and inappropriate behavior. We've decided this as a group. And our brains then develop to meet these standards that we've set. But we can also deconstruct the brain into connectomes, or macrocircuits and microcircuits and neurons and synapses and molecules, and finally DNA. And what we're trying to accomplish this week is to talk about all these levels of organization. So we're going to have people talk about development. We're going to have people talk about molecular mechanisms involved in epigenetic changes to the brain. We're going to have people talk about social interactions among human primates and non-human primates. We're going to have people talk about the evolution of complex behaviors such as language. So some of the questions I hope that we're going to be able to address this week: what factors contribute to brain organization, connectivity, and function during development and in species over time? Because the two interact completely. How do complex behaviors develop within an individual? How are complex behaviors such as tool use and language perpetuated within a population? Is it genetic or is it epigenetic? To what extent does human social evolution impact biological evolution? And I'm pulling these apart as if they're two separate things, but they are not mutually exclusive. What are the underlying mechanisms that generate these phenotypic alterations at all levels of organization, from cellular to systems to behavioral? And how do higher levels of organization, such as social systems, emerge from lower levels, such as brains composed of neurons? And what are the boundary conditions at each level, which is very important. What are the constraints? So just very briefly, I want to introduce our speakers. And this is for the students so that you can actually know what the people look like. And we want you to interact with our speakers. I don't think Paul mentioned this, but lunch is a really good time to sit down with the speakers. Don't be afraid to park yourself right next to them and say, I liked your talk, or I know your work, or that was really fascinating, can I talk to you? Because what happens in past years is the students kind of segregate to themselves and they feel that maybe they shouldn't interact with the speakers, but we really, really want to talk to you. And it'll be good for you too, because you'll say, hey, they're not as smart as I thought, and and I can do science if they can. Okay, so I want to introduce our and then I'm going to give it. This is just my little introduction, guys, and then I'm going to give a talk after our break. Okay, so our first speaker, one of our first speakers today is going to be Dr. John Cause at Vanderbilt University. He's in the Department of Psychology, and he's going to talk about the evolution of the human brain, a systems-level comparative approach. Tomorrow we'll have Zoltan Molnar from Department of Physiology, Anatomy and Genetics at Oxford University, which I forgot to put in there. He's going to talk about evolution of cerebral cortex development, some of the stuff that Tony talked about. We have Danielle Stolzenberg, University of California, Davis, Department of Psychology, learning to mother, epigenetic mechanisms in the maternal neural circuit. I'm really excited about some of this epigenetic stuff, and I've tended to focus a lot of speakers on epigenetics because I think this is the new really important thing, not the boondoggle like big data, but the new important thing in neuroscience is epigenetic mechanisms. Dorothy Ferghese, who's going to be here on Wednesday, University of Georgia, Department of Psychology, tugging at tool use. She does really, really cool stuff in non-human primates, in Cebus monkeys, looking at cultural transmission of tool use of tools in the wild, experiments in which, and experimental situations in Cebus monkeys. Asif Ganzafar from Princeton University, Department of Psychology, another great speaker. He's going to talk about the developmental neuromechanics and evolution of vocal cooperation in non-human primates. Gary Marcus will give a talk, I think, on Thursday. Gary, are you here? I guess he sent an urgent message, how do I get to the meeting? Obviously, he can't read maps. He's from New York University. He's going to, Department of Psychology. He's going to talk about bridging biology and computation in the service of understanding how language evolved. I think it's going to be fantastic. Henry Kennedy will be talking on Thursday. So some of the speakers are here and some of them aren't here. But they'll be coming throughout the week and overlapping on several days. Stem Cell and Brain Research Institute in Lyon, France, The Brain in Space. Murray Shanahan, Imperial College of London, will be talking about metastability in neurodynamics. Randall Beer, Indiana University, Department of Cognitive Science, Evolution Analysis of Brain, Body, Environment Systems. Okay, so Jackie Setti, Max Planck Institute for, I'm going to just explain this, for Informatics, Computation, Systems Biology, in silico models of stem cell and developmental system. Now, I invited some of the speakers this week, and Tony invited some of the speakers, and I thought I was going to introduce my speakers, and Tony led me to believe that he was going to introduce his speakers. So last night at 1 o'clock in the morning, we're sitting around my table, and Tony's like, well, no, put pictures of my speakers in there too. So I got online. And I'm like, well, what does your speaker look like? Because I don't know. Which is also cool. A lot of these speakers, some of these speakers I never met some of the speakers that I invited, but I liked their work. So that's why they were invited. I invited people that I wanted to hear give a talk. So we got online, and one choice was, this could be Jackie Setti. And I'm thinking, no. Another was, this could be Jackie Setti, but I don't know if it's going to be old enough. This could be Jackie Setti, and I will say something about Jackie Setti, and we should all ask him this. How has this man managed to keep his picture off the internet? Which one is it? Man, you say. Well, I don't know. It could be Jackie. Is it a man or is it a woman? Right? So, how has he managed? I mean, I get online, and there are so many.
How do we now avoid imposters?
We can't. It's impossible.
So Jackie Setti can be chosen.
Who knows? Is there a Jackie Setti, right? So, no, I mean, I get on the internet, there's so many horrible pictures of myself that I never put on there. How could this man have managed to actually never have his picture associated with his, I mean, that's extraordinary. That, he must be a really smart guy. I can't wait to hear his talk. He's just a brain. So we'll see if any of these people are in fact Yakiseti. If anyone knows what he looks like, I would appreciate a picture. But this is what you come up with at 1 o'clock in the morning after several glasses of wine and saying, oh my God, I got to go to bed because I got to give a talk tomorrow. Okay. So discussants, we're also going to have discussants, and they're going to be giving some tutorials. Unfortunately, I just got an email from Cedric Boakes. He was supposed to be a discussant, and he said he's not. He can come to some of the talks this week, but he has a heavy semester, so he may not be able to participate as a discussant, which is unfortunate, but hopefully we'll see him. We have Stuart Wilson from the University of Sheffield, Department of Psychology, Simulations and Computational Models. Danielle Stolzenberg is also going to be giving a tutorial on epigenetic mechanisms. Scott Simon, University of California. I could not find a picture of Scott where he did not have a beer in his hand. This is worrisome, especially since he's my husband. Biologically inspired structural designs. And microfluidics. It's all about fluid. That's what he's telling me, or tries to convince me. Okay, so I'm going to end there so you know what the speakers look like. I think we're taking a break right now, and then we're going to move on to, is that right? Yep. Anna? Yeah. And what time is it now? What time do we have to be back?
From 11 to 11:30 we have a coffee break.
Right, so is it what?
11:30 we're back.
11:30 we're back.
Yeah, so 11:30. And guys, don't like mingle too long and like sort of straggle in. Be here at 11:30.
Very good. All right, Leah, thank you.
Okay. Talking about, or this week we'll be talking about, genes that are involved in aspects of development that generate certain aspects of a cortical phenotype. Then there are activity-dependent mechanisms, which I'm going to focus on mainly today, that generate a particular type of phenotype. And a long time ago, the titles of my talk used to be, How Does Evolution Build a Complex Brain? And I realized that was very, very limiting, because if you talk about evolution, you're talking about genes, and there's so much more to the story than just genes. So I've changed it to focus on the cortical phenotype, and I am going to talk mostly about newer work in my laboratory, done by some of the students in the audience, and how early experience can actually change the structure and function of the brain in a very, very dramatic way. Oh, so also, I don't have a brain on my first introductory slide. These are a bunch of different rodents, and I really like this slide because, as I said earlier, you can't just talk about the brain. You really have to talk about the body morphology, locomotion, special effectors like whiskers, special morphological specializations like wings or the ability to glide, to look at how the brain organizes itself and how important this is in the development and evolution of the neocortex. So this is a back view of a dolphin brain. And this is a little South American marsupial. It's a native cat. And you can see the brains vary dramatically in size. These aren't even taken to scale. You have this massive expansion of the neocortex in a dolphin and a little tiny cap of neocortex right here in the Australian marsupial. So why am I interested in the neocortex? I'm interested in the neocortex for the reasons that Tony mentioned. It is a portion of the brain that is involved in complex behavior, such as learning, perception, cognition, language, reading, writing songs, etc. It's also the portion of the brain that's changed most dramatically in mammals compared to other portions of the brain, and it is particularly large in humans. And it's not just that the brain has gotten very large, and we've known there's a correlation between the relative size of the neocortex and what we'd like to call intelligence or complex behavior. But we've known for a long time, this is a classic work of Corbinian and Brodmann back in the early 1900s, where he cut the brain and what are the background of people here? Psychology, engineering, robotics. So I'm just going to assume that people don't know about brains. So I'll say this is a brain. Getting real basic. This is the front of the brain. This is a dorsal view of the brain. So this is a dorsolateral view of the brain. And what he did is he cut the brain and cut lots of brains. He stained the cells, and he saw that the neocortex was not homogeneous, that it was actually composed of different areas, architectonic areas, he called them, that looked very different. And that humans had a lot of those areas, so the different stippling represents these heterogeneities. And if you looked at something like a European hedgehog with a little tiny cap of neocortex, it had very few areas. He proposed, even before he stuck an electrode, before an electrode was ever stuck in a human brain or an animal brain, that these were correlated with functional areas, and that different portions of the neocortex are involved in different functions or processes. He also noticed in this comparative analysis that there were certain regions of the brain, this is the primary visual cortex back here, we know now, or area 17, that certain regions of the brain looked the same in all these species and probably were homologous or inherited from a common ancestor. So the question is, fossil studies and also comparative studies, like Tony said, on Monodelphis domestica, and I don't have the largest colony of Monodelphis domestica. Southwest Biomedical has like thousands of them. But in terms of a personal PI colony, like I have a personal colony. for a variety of uses, including science. We know from fossil records that the brains of early mammals were very small. They had a little tiny cap of neocortex. I've just color-coded cortical fields. It's not important that you know the names of them, but I'll just say the blue represents a primary visual field, primary somatosensory field, primary auditory field. So they had a few brain areas, maybe 10 or 15 brain areas. And we have species like primates, and this is certainly a very simple rendition, have many, many more fields. And the question is, how does the brain evolve from a simple form with few parts to a complicated form with many parts over a 2 million year process? So there's a problem. So that's a great question, but there's a real problem with that because it happened over 200 million years. And these changes take generations. So I can't really study evolution directly. I don't have 10 million years to sit around and see how the brain changes. And if I did, I bet I'd be doing something way different than that. So how do we approach this problem? And this is the Evo-Devo part. We can study the products of the process. What does evolution produce? What kinds of brain does evolution produce? So starting when I was working in John Caz's laboratory, and still, we still do a lot of comparative analysis. I can look at a lot of different brains, and I can say what's the same, inherited from a common ancestor, what's homologous, and what's different. So what changes have evolution produced? And what we found is that evolution, in my opinion, is the continuing process of limiting our options. So that if you look at a variety of different mammals, from monotremes to primates, there are certain features of the brain that are the same. And the ways in which brains have changed at a systems level is highly restricted. But this is only looking at frozen moments. So I'm taking a snapshot of evolution of a species and within the life of an individual, and I'm stopping it and I'm looking at it. I'm taking another snapshot, another snapshot, and another snapshot. The brain has changed. What we don't know are how those phenotypic changes have occurred. What are the transitions? How do they occur? Evolution is the evolution of the neocortex is actually the evolution, as Tony mentioned, of developmental mechanisms that give rise to aspects of the cortical phenotype. So studies of development can show us how changes have occurred in species over time. So the methods that our lab and other people have used, and we are going to be hearing speakers that are going to be talking about probably all of these types of methods. Anatomical, looking at connections of the brain, or how the brain actually looks when you stain it with different stains. Physiological, so if I stick an electrode in the brain, what do neurons want to listen to? Histological, the appearance of the brain. Molecular, genetics, behavioral, social. And it allows us to segregate the neocortex into a variety of parts. And we know even from work, even before we did work on humans, animal research has shown us that there are portions of the brain that are involved in visual functions, somatosensory functions, auditory functions. And then in humans, we have prefrontal cortex or posterior parietal cortex involved in more complex functions. So comparative studies in my own laboratory and a variety of other laboratories, including John Caz's, have allowed us to generate this phylogenetic tree, or this cladogram. These are the brains of a variety of different species, including marsupials, monotremes, marsupials, primates. And the patterns are similar cortical fields or homologous cortical fields that have been retained from a common ancestor. So even though we can't really know the brain of a common ancestor, the most parsimonious explanation for all of these species having a similar form of organization is that it was inherited from a common ancestor and didn't evolve de novo in every single lineage. So also really important, very important, that this plan exists with the primary visual field, the primary somatosensory field, primary auditory, even in the absence of use. So one of my favorite examples is a blind mole rat. It's got little micro-ophthalmic eyes, so its eyes are tiny. Skin has grown over the eyes. That animal still has a primary visual cortex. It still has a geniculocortical pathway. It's been co-opted by the auditory system, but you can't get rid of it. So there are clearly some constraints. So the brain can't become anything. There are clearly constraints on how you can build this complex piece of tissue. So there is a similar plan of organization that all species have. I share a common plan with a monotreme and a mouse, but there are clear differences. So this is a flattened macaque monkey brain. This is the front. This is the top. This is a flattened mouse brain. All of my illustrations will be in this sort of format. Unfortunately, some of them are facing different ways. And what we see is, here's my plan in red: primary visual somatosensory cortex, primary auditory cortex, primary visual cortex. But there are huge differences in this brain compared to this brain. We have changes in the size of the cortical sheet. We have an addition of cortical fields. We have changes in the relative location of fields, and so on. And this is a little schematic I made some time ago and have reused many times to show you these general systems level modifications. So if I do this large comparative analysis, I look at a lot of different species, and I say, well, what sorts of changes have been made, systems level changes have been made to the brain? And this is supposed to indicate a cortical sheet. These are individual cortical fields, so we have changes in the size of the cortical sheet that we see in different species, sometimes independently evolved. So humans have independently evolved a large neocortex, as have dolphins and elephants, for example. Sensory domain allocation, this is really important. So, for example, in somatosensory cortex, you have a representation of your contralateral body, of your cutaneous receptors, of your deep receptors, muscles, joints, and so on. Depending on how I use that part, I can have a magnification of that representation. So in humans, there's a magnification of the digit tips. There's a magnification of oral structures and so on. In something like a mouse or a rat, where they are exploring the world and using their vibrissae as major effectors, they have a very large representation of the vibrissae. So that's what I mean by that's what I mean by magnification of behaviorally relevant body parts. Sensory domain allocation. So if I'm an echolocating bat, I have a ton of cortex devoted to auditory processing. And if I'm a primate, a highly visual primate, I have a lot of cortex devoted to visual processing. That's sensory domain allocation. There have been changes in the relative size of cortical fields. So while I have a V1 and a blind mole rat has a V1, relative to the size of the cortical sheet, the blind mole rat's V1 is tiny. Mine's much larger. I already told you what I mean by magnification of behaviorally relevant body parts. Addition of modules, which I'm not going to talk about. I already mentioned number of cortical fields, and I think very, very important, and probably the thing that distinguishes, in my opinion, humans from our close relatives, chimpanzees, are changes in the connections of cortical fields.
Taking questions along the way?
Oh, yeah, sure.
I feel like missing is maybe H, rewiring within a particular cortical field. Maybe that relates to the E that you didn't talk about.
Well, so I would say alterations in connections can be between cortical fields, or you're saying within a cortical field itself, so you have alterations in intrinsic connectivity, and you do. Yeah, and are you going to talk about that at all? Oh, sorry. Yes, that's there too. And I won't say this list is exhaustive, but looking at a lot of different species, I wouldn't say there are probably many, many more types of changes.
I mean, I think these are all important. I feel like H is neglected in the field because we don't know enough about how to characterize it, but it might turn out to be really important.
Well, actually, at the end of my talk, I'm going to show you individual differences in voles based on early experience. And one of the things we see are differences in intrinsic connectivity within a cortical field. So the premise is that natural variation of these cortical organizational features within a population must ultimately lead to speciation.
But Leah, another question. So do you really need to distinguish modules from fields?
Well, it's a great question, because a cortical module, so the classic idea of a cortical field is it looks a certain way architectonically. It has a specific set of connections. If you record from neurons, they respond to a very specific type of stimulation. If you lesion it, you'll have some specific deficit in behavior. But modules actually follow all those rules as well. Mm-hmm. And they are embedded within a cortical field. So I believe that that's a kind of slippery slope. That you would
be happy if we would all call it fields, for instance?
We can all call them fields, or we can call them modules. And there are some larger modules that are more heterogeneous. And then there are I'm talking about the smaller things that can be embedded within a field. So last year's talk, I think modules are important. I think that what they represent sometimes is a cortical field embedded within a cortical field. But they follow, the criteria that we use to define a cortical field. That's a really good question. So what I'm going to focus on today is sensory domain allocation, magnification of behaviorally relevant body parts, and connections of cortical fields. So a question, very important question, is what factors contribute to these phenotypic modifications? There are things that are intrinsic to the neocortex, so genes in the developing animal, and Zoltan Molnar is going to talk about that tomorrow. So I had a segment of the talk that I used to talk about that, but he's going to do a much better job than I can. Things extrinsic to the neocortex. So by extrinsic to the neocortex, there are two types of things that can be extrinsic to the neocortex. Ones are genes that regulate body morphology. And body morphology, you're going to see, plays a really important role on how the neocortex develops. But the other thing that's extrinsic to the neocortex is the environment, or sensory-driven activity. And Danielle's going to talk about epigenetic mechanisms that can modify the phenotype. So we've done for years and years and years, we did this comparative analysis, came up with this nice set of here are the types of things that change. It's hard to come up with theories of cortical field evolution, but one way you can test how these modifications may have come about is we say, can we induce these systems-level modifications in a developing nervous system and generate a phenotype that is consistent with what evolution would produce? So in the following talk, I'm going to sort of intersperse examples from the natural world, some comparative stuff from different species, with experimental manipulations of the developing nervous system, experimental manipulations in peripheral morphology, experimental alterations in sensory-driven activity, and then I'm going to go back to natural variation and early sensory experience within a population and see if we can induce some of these changes to the phenotype, such as changes in connectivity, changes in sensory domain allocation, changes in function, and so on, without changing anything intrinsic to the neocortex itself. So changes in peripheral morphology, this is my favorite observation in the whole of the natural world. I really mean that. That sounds very dramatic, and like it is my favorite observation. And this is my favorite observation. This is a duck-billed platypus. I was working in John Koz's laboratory, and I was all set to go to MIT. And I said, I don't really want to go to MIT. I know I should go to MIT and get my PhD and be, MIT educated. And be a card-carrying neuroscientist. But he said, well, what do you want to do? I said, I want to go to Australia and study duck-billed platypus. And he said, who do you want to work with? And I said, I don't care. I just want to go and study the duck-billed platypus. And he was mad at me for about 2 weeks, and then he thought it was a good idea afterwards. I talked him around. So this is the duck-billed platypus. It's a really beautiful animal. It has a specialized effector, which is a bill. And on the bill, you have two types of receptors. You have mechanosensory receptors aligned in a line like this, and then electrosensory receptors, and they're interdigitated. When the animal does anything that's important, such as eating, mating, hanging out with other platypuses, it closes its eyes, its ears, and its nose. So it is one big, huge, enormous bill. And it's using electrosensory reception to navigate large distances and capture prey that are moving their muscles. And it's also using its mechanosensory system. And if you look at the neocortex of this species, it's extraordinary. This is the front of the brain, the front of the neocortex. This is the dorsal portion of the neocortex. And this is a platypunculus, or representation. I made that word up, and I do that all the time because I'm a neuroanatomist, and you're allowed to do that. And people look at it and say, I'm going to go to the dictionary and check that word out. So this is a platypunculus. What I want to point out to you for people who aren't aficionados in the neocortex is that if I looked at the amount of the neocortex that's representing the bill, this is the entire cortical sheet. Here's the bill in the primary somatosensory area. Here's a bill in another field called the rostral field. Here's a bill in the caudal field, which is S2 or PV. This is the representation of the body, this little tiny thing right here. Auditory cortex is completely embedded. So about 75% of its entire cortical sheet is devoted to processing inputs from the bill. It's amazing. And then if you look at the distribution of electrosensory and mechanosensory, you have this interdigitation within the cortex itself. So you see this huge sensory domain allocation. So most of the cortex is devoted to processing inputs from the somatosensory system. And you also see this magnification of behaviorally relevant body parts, like the bill.
What's the purple area?
Which one?
Responding to?
This one right here?
Well, that looks a bit like the body map.
This is a body.
But you were pointing to this whole area C and below.
Oh, this is bill. This is bill.
Okay. Which part of the bill? Because the red stripes, I guess, respond to the red stripes you have in your figure. So the purple guys are interdigitated.
Right. So these are interdigitation of mechanosensory and electrosensory inputs. So the light blue is electrosensory and the red is mechanosensory. this is all, this was, when we mapped this, we did not look at if it responded to electrosensory stimulation. We were just doing the somatosensory map. So there may be more electrosensory maps. But this is what I'm saying. If you look at this this is your module question. This would be a module. But if I took all these red parts and put them together, you would have a complete representation of electrosensory receptors or mechanosensory receptors. And if I took all the light purple parts and put them together, you'd have a complete representation, right? So.
But another question. So if you look at electric fish, there a lot of these electro-sensing systems projected to the cerebellum, because this might give you then, let's say, a high temporal resolution of processing. Is it the same for platypus?
Don't know. So what happened was it was really hard to work on platypus. I had to go to the government offices and get permits. It took 9 months. I was able to get 3 a year, then 6, and we built a platypusery, and it was a huge deal. I know that's a word. It sounds naughty, doesn't it?
That's a concept that might get you in trouble.
Got arrested 5 times. Okay, so the question is this huge change in the neocortex, is there something intrinsic to the neocortex that's generating these types of maps, this sensory domain allocation? Is it simply that there are changes in the body morphology that then manifest as these sorts of changes in the neocortex? So I love this work. This is by, I never met this guy. I'd like to invite him maybe sometime too. Kritos. So if we look at adult forelimb morphology, this is a morphology of a mouse. I love this picture. This is morphology of a bat. And if you guys are not familiar with bats, this is the forelimb of a bat. These are the digits of a bat. They've undergone dramatic changes. This is the thumb. They climb like this. So the fingers have elongated. The bone density has decreased. They have interdigit membranes. Very, very different. And it turns out if you do comparative analysis of forelimb development, what you see are that this happens to be one of the main players in forelimb development, PRX1, expression of PRX1. It changes very slightly in a bat versus a mouse. And this, along with a few other genes, just changing their distribution slightly, and Tony, you talked about this, can produce radically different peripheral morphologies. Now, and there are specializations on the wing that are called touch domes that are exquisitely sensitive to small changes in air pressure. And of course, bats have a very large auditory cortex, but if you look at the forelimb representation in bats compared to a mouse, you have this really large forelimb representation in bat. Is that due to something intrinsic to the neocortex, or is it simply due to a change in peripheral morphology that manifests? So we'll do some experiments to test if changes in peripheral morphology can generate these phenotypic changes to the neocortex. A good important point is that all brains follow the same rules of construction and operate under the same constraints. So I showed you the example of the duck-billed platypus and this huge representation of the bill, taking up most of the neocortex. Ken Catania has done beautiful work in naked mole rats where they have specialized incisors that have an enormous representation. He's also done beautiful work in the star-nosed mole. We see this similar principle of sensory domain allocation and certainly magnification of behaviorally relevant body parts. We like to think of humans as somehow special or stepping outside the bounds of evolution, but we can reinterpret Broca's area of specializations of the oral structures, the changes to the larynx, tongue, palate, and so on, as specializations of premotor, motor, and also somatosensory cortex, like we see in all other species that have undergone changes in connectivity, et cetera. Okay, so this comparative observation led us to do several experimental manipulations of peripheral morphology. And the first one I want to talk about is bilateral nucleation in Monodelphis domestica. So the reason we like to work on Monodelphis domestica to do our behavioral experiments, mice you can do genetic manipulations, highly targeted. These guys, what we do is we do physical manipulations. So this is a P4 Monodelphis. You can see it has a little hind limb bud. It's got a little forelimb. It's attached to the nipple. It only has a little eye spot. Very important for us, the development of its brain is really immature. So at P4, the thalamus, thalamocortical afferents, the structure that's carrying information about peripheral receptors to the neocortex, those connections have not formed. They don't form until about P7. The eye has not, retinal ganglion cells from the eye have not grown into the diencephalon, so this brain is not connected up. It's very, very immature. So what we do is we remove both eyes, or what will be the eyes, the little eye spots, at P4, so at a very immature state. We let the animal grow up, and then we do electrophysiological recordings of its neocortex. We look at cortical connectivity. We look at architectures to see, okay, what happens to visual cortex? Am I going to have a visual cortex? Will it be like a blind mole rat? And so we do several changes that we can measure. So what we found is that there's no change in the overall size of the cortical sheet. And I should say this is the front of the brain. Here's the top of the brain. And when I say cortical architecture, what's very nice about these guys is even if you guys are studying robotics or engineering, you can see that this is a highly myelinated area. This is very light. And this turns out to be the primary somatosensory area. Here's the primary visual area, second visual area, auditory cortex. What's really cool is that in bilateral enucleates, they have no eyes. They still have an architectonically defined visual cortex. It looks like visual cortex in the normal animal, but it's really small. They have a somatosensory cortex. If you make measurements, what you find is...
How do you know that that structure is a visual cortex?
Well, I said that you can identify fields histologically. So when this is stained for myelin. So we say it's visual cortex because it looks, it's highly myelinated like visual cortex, but it doesn't get input from the eye. It's also, I'll show you, it still gets connections from the lateral geniculate nucleus, even though the animal doesn't have eyes. So it's very much like the blind mole rat. You can't get rid of this, but I shouldn't call it V1. It's not visual because there are no eyes, but it looks like V1 in histologically prepared tissue, and it has some connections that are similar to V1. But let me just continue this a little bit, and if you still have a question, we'll get back to that. If we measure somatosensory cortex, what we find is that somatosensory cortex has expanded in size and visual cortex has shrunk in size, or what should be visual cortex has shrunk in size. If we stick electrodes in the neocortex, so blue dots, so each one of these dots is a place where I've stuck an electrode in the neocortex, and I'm saying, what do neurons want to respond to? Do they want to respond to visual stimulation, somatosensory stimulation, auditory stimulation? So if they respond to visual stimulation, it's marked in blue. If they respond to somatosensory stimulation, it's in red. And if they respond to auditory stimulation, it's in green. This is a normal monodelphis domestica. Somatosensory cortex responds to somatosensory stimulation, visual cortex, visual stimulation, auditory cortex, auditory stimulation. There's some multimodal cortex in here. In my bilateral enucleates, if I go back into what should be visual cortex, neurons obviously are not responding to visual stimulation, but they're not just doing nothing. They're not just hanging out. They're responding to somatosensory stimulation and auditory stimulation. Some are mixed. And I want to point out that in this case, somatosensory cortex is neurons responding to somatosensory stimulation. In this case, it's responding to somatosensory and auditory. So somatosensory cortex has changed. And we saw a lot of variability in these guys. And I can tell you why I think that's so, but that could be later. If I look at what body parts are represented in that new visual cortex that's no longer visual cortex, I see that most of the receptive fields are on the face and the snout. So you have this huge expansion of the snout representation in the neocortex. So without doing anything to the cortex itself, I've changed the functional allocation so that all of what would be visual cortex is now somatosensory and auditory. I've changed magnification of behaviorally relevant body part. And if I place injections in what would be visual cortex, this is a normal animal, and normally visual cortex just projects to other visual areas, this other area called CT and entorhinal cortex. But if I look in the monodelphis domestica, in area 17 in the bilateral nucleate, in this area that looks like area 17, like normal visual cortex, but it's smaller, what I see is that I'm getting input from somatosensory cortex, I'm getting input from auditory cortex, from frontal cortex, and if I look at the thalamus, I'm getting inputs from somatosensory nuclei of the thalamus. So all I've done is completely eliminate all visual input really early in development before the brain has hooked itself up. And what I've done is I've completely changed the functional organization of the brain and the connectivity of the brain in a massive way. Okay, and that's just a summary of what I just showed you. Normal connectivity, and then we have all these other inputs to this new field. But I said, what was interesting, and oftentimes when people are saying, okay, I want to perturb some aspect of some sensory system, they're only looking at the sensory system that they've perturbed. They're not looking at what's happening to the remaining systems. So we noticed we had variability in our functional maps, so that in some animals you had, somatosensory cortex was responding to somatosensory stimulation, but it was also responding to auditory stimulation, which you don't see in normal animals. So,
since this is not seen in other animals, those animals that do have this behavior, do they show any special?
Great question.
Behavior?
That's a great question.
Enhanced behavior?
That's a great question, and we've been. Deepa's in my laboratory, Jimmy's in my laboratory, and I've been trying to get behavioral stuff going in monodelphis domestica for a while. One would expect what's the point of having this enlarged representation of the vertebrae? You would expect them to be super tactile animals compared to, and so we don't know, but it's something that we're pursuing, but it's a great question. And I'm not going to talk about it today, but the other thing is, can I direct this natural plasticity? Because sometimes it's auditory, sometimes it's somatosensory. If I rear them in a highly tactile environment, if they have no visual inputs, and rear them and push it towards the somatosensory system, can I make them super tactile individuals? And I think yes. I'm going to show you some of that later in this talk. So, okay, so this is work. So we're saying, okay, so what about, let's study the connections of somatosensory cortex. And this is work by Jimmy Dooley in my laboratory. It's in preparation. So what he did is he looked at the connections of monodelphis in normal opossums, and is connected with other somatosensory areas, a little bit with frontal cortex, medial wall. And he looked at the connections of somatosensory cortex in bilateral enucleates. And what we see are really huge changes in the connectivity of the spared sensory system. So it's not just that what would be visual cortex is reorganizing, somatosensory cortex is reorganizing as well. So how information is being processed is radically different in these animals compared to sighted individuals. So you're seeing connections with a lot of connections with multimodal cortex. Actually, here's a nice overview. And these are just a couple of ways to show this, but in normal animals, most of S1 is receiving input from other somatosensory areas, and you have a little bit from other areas and a tiny, tiny bit from multimodal cortex. If you look at the distribution of sensory modalities that are projecting to or cortical fields that are projecting to S1 in the bilateral nucleate, you could see very, very large differences. Okay, so experimental manipulations on sensory-driven activity. So that's, I mean, you're pretty much taking a hatchet to the system. I'm removing both eyes. That is a huge manipulation, right? That doesn't happen in evolution. Some people are born congenitally blind, and if they're born congenitally blind, then they have a massive reorganization of our cortex. So if you imagine, and I always try to imagine these things. It's going to sound weird. I imagine what it's like to be a platypus, but I can't, right? Because you see what's going on. But I can't even imagine what it's like to be blind. How could I? Because it's not just I'm going to close my eyes. Obviously, what we're showing in monodelphis domestica is happening in congenitally blind individuals as well. They're having a major rewiring of their sensory cortex. And it's going to be dependent on early sensory experience, which can be different. I'm going to show that a little bit later. Okay. Anyway, so enough of what I imagine what it's like to be. There are a lot of other things I imagine what it would be like to be. Okay, anyway. A German showgirl is one of them. Okay, it's a private joke. It's a private joke. Okay, so I'm taking a hatchet to the system, right? How about if I just manipulate sensory-driven activity? Can I alter, if I alter the physical environment in which the animal develops, so not alter the body itself, but the physical environment, can I change cortical organization, cortical field size, cellular composition, neuronal response, properties, connectivity, and behavior? So without touching the morphology or the cortex. So this is work in our laboratory where we reared animals in what we're calling an enhanced environment. It's just basically a different visual environment where it's being reared in a cage of stripes. Look, this is also work from Jimmy in progress. So he's looking at response properties of visual neurons and visual cortex. And what he's showing is that this is in normal animals, looking at the highest spatial frequencies that neurons will respond to, looking to see if neurons are oriented or not. Most of them are not oriented. And these are in normal animals. And the hypothesis is if we rear them in a striped environment that's a higher spatial frequency than what they can normally see, that we're going to push their spatial frequency higher so that their acuity is going to be better. And neurons will respond to those spatial frequencies. So this is the predicted change in terms of spatial frequency and orientation selectivity. And what we showed is that we didn't find that, but we did find differences. It was really interesting. What we found is that they actually, most neurons responded to a lower spatial frequency. This is the control is blue, striped reared is red, a lower spatial frequency than what we would expect, and that they had a preference, neurons had a preference for cardinal orientations. One possibility, and we were sort of looking into this, is that we didn't consider viewing distance. If they were viewing up very, very close, then they would be experiencing larger spatial frequencies as opposed to viewing the stripes from further away. But what was really cool is that, we didn't expect this, but this is where we started doing some behavioral stuff, and Deepa came to the laboratory, and we were just doing some really crappy behavior, and she really helped us improve our system. This is a cortically mediated behavior. It's called the jumping test. And what you do is you train the animals to jump to the stripes, and you can change the distance between the stripes and make them higher and higher spatial frequencies. And if the animal jumps to, if it can't detect the difference between a gray and the stripes, you've determined its acuity. And you asked about behavior, and I'll tell you, we're doing behavior in our laboratory. It's the hardest thing I've ever done in my entire life, because the animals tend to train you. You're all trained up. You know what size crickets they want to eat. You know what kind of lunch meat they like. But they'll maybe do a couple of, you can ask Deepa about it. But she's done this, and I just want to show you this because it's pretty nice. And this is what the test looks like. And there's a platform in the middle, and the animal's looking and checking things out and sniffing. And it's pretty much like watching paint dry. Yeah, there you go. And he gets his treat. So we determined the acuity. And what was really interesting is in the striped-eared animals, it turns out that their acuity is less than normal animals, sort of going along with what we saw with the neurons. Again, we think it probably has something to do with viewing distance. Okay.
Another question, Leah. Look, the projections from the thalamus to the visual cortex are in themselves also bandwidth constrained. They're not like just random.
Right.
Right? So isn't that already, if you want, restricting the kinds of changes you can expect?
Yeah, so the point is, can the cortex become anything? No, it can't. And in fact, there are some constraints, but it can become a lot. So what this says, what certainly the bilateral nucleate stuff says to me is that thalamocortical afferents are really, really exuberant. And they get pruned back, but they're not so exuberant that you can change the back of the brain into the front of the brain. I mean, a lot of these experiments is I want to push the cortical phenotype and see how far I can push it. But what's extraordinary is you can push it really, really far.
But given the modulation you've got, you must be able to predict the mean distance the animal was at. Don't look at your watch, because you have a lot of time. The mean distance.
No, I hate people who talk like forever.
Yeah.
And it's like an hour. It's like, okay, I don't.
Terrible,
It's just like, shut up.
Exactly. But look, let me finish. The point is that given the modulation you've got, you must be able to predict the mean distance to your stripy pattern, right? Because that would be then the viewing distance which you have to size that.
So we had people look at. Go ahead.
I was going to say we've done some of that, and the hardest part is then, because we're just putting these animals in their cage, is, if it's in the corner and it's facing, what's it looking at? When is it actively looking? When is it, its eye closed and it's just smelling things? We know it spends most of its time in the corner, but it's probably not like looking and attending to the complete opposite wall. So, we designed it with a spatial frequency well above what the normal spatial frequency of the animal is. But it's really difficult by just tracking the location of the animal to get when it's paying attention to visual stimuli. We could say it's at the maximum viewing spatial frequency from the opposite corner or below, but what the exact composition of that is very difficult to determine.
Okay.
And like the most boring video in the world to watch. It's amazing. I have any graduate students who want to work in the lab at all. Okay.
Don't tell them that.
I know. Natural. I'm going to show you some of these experiments which are really cool, which I talked Katie Campion to doing. So natural differences in sensory-driven activity. These are comparative studies in rodents. I love these studies. Okay, so here's some rodents. This is a Norway rat. Here's somatosensory cortex, visual cortex, auditory cortex. This is a Norway rat that was caught in the wild. So same species, except it's living out in the wild in Davis. Nile grass rat. A California ground squirrel. Eastern gray squirrel. What I like about these four animals, or sorry, this one, is they all live in Davis. Two are diurnal. One's living on the ground, one's living in trees, and one is nocturnal and terrestrial, but climbs trees too. So I was able to talk Katie Campy. I said, hey, wouldn't it be a great idea to go out and catch some wild rats? She said, yeah, this will be fantastic. I mean, it was amazing. And they're mean. Ooh. And they carry the plague. But she was right into it, right? So I'm very persuasive PI. So what I want to show you, so these are the same species. Oh, and she did a couple of measurements. She did some in the squirrels as well, but I'm not going to talk about those. So this is the blue is the laboratory rat, and the red is the wild-caught rat. And what she saw was that in auditory cortex, the laboratory rat had a significantly larger auditory cortex, probably because that sense was less impacted. And that somatosensory cortex was significantly larger in a laboratory rat than in a wild-caught rat. And this is only the tip of the iceberg. Unfortunately, we didn't do connections. But we also did what's called the isotropic fractionator method, which allows us to look at the density of neurons within a cortical field. So this is a DAPI, and you just take a piece of cortical tissue. This is a DAPI stain, which stains all nuclei. Then you can stain a subset for neurons, and you count the proportion of neurons to non-neurons within a given piece of tissue, and it allows you to calculate a number of interesting aspects of that piece of tissue. So this is laboratory rats are in blue, and wild-caught rats are in orange. And what we find is if we look at the percentage of neurons, so we take out area 17, grind it up, stain it for DAPI, do the isotropic fractionator method, and this indicates significantly different. So they have a larger percentage of neurons. Wild-caught rats have a larger percentage of neurons in visual cortex, and they have a significantly larger neuronal density in visual cortex as well. So we didn't do any experimental manipulations, but it's not surprising. I mean, it would have been great if we could have done connectivity, which would have been fantastic, and some electrophysiology, although, there's still more Jimmy, Deepa. I'll have to get another graduate student to go out and catch some wild-caught rats. But there are very, very large differences, and it's not surprising because you have an animal that's being reared in a very restricted environment versus one that's out in the world doing more natural things. Okay.
One second. If the changes in the neuron numbers, right, there must be also some sort of genetic component to it. Or you think it's really all epigenetic, or it's all dependent?
They're the same species. So they're the same species. So that's the idea, is that I think it is all epigenetic. And if you let me give this last part, I'll tell you why. Because these are the coolest experiments on the planet.
All right, cool. Let's get to it then.
All right. Okay. So natural differences and this is done in collaboration with Karen Bales in my laboratory, and it's also work of Adele Selke. Natural differences in rearing conditions, and this is really awesome, can generate changes in a lot of aspects of the cortical phenotype. And these are prairie voles. And I don't know if you guys are familiar with prairie voles, but they are monogamous, and they're biparental, meaning that both the father and the mother help rear the offspring. And what's really interesting about these guys, and they've got this really interesting perioral face region. These are the lips. The hairs actually go inside the mouth. Is that you can quantify, so you have both parents are rearing the offspring, and you can quantify the amount of tactile contact that prairie voles have. And it turns out there's a normal distribution of parental rearing styles, with some individuals, some parents having very, very high tactile contact. So you can measure the amount of time they spend grooming, licking, nursing the young, and some having low contact. And one is not better than the other. It's just a difference in the amount of tactile contact that the individuals are exposed to. And this can be measured, and it's significantly different. So high contact is in red, so things like non-huddling contact, neutral nursing, lateral nursing, low contact. And if you look at parental care, maternal and paternal care, significant differences in total contact time. And what's really interesting is Karen and Alison Perkinbilly have done some cross-fostering work, and it turns out that if you're from a high-contact group of parents, you become a high-contact parent yourself. If you cross-foster, meaning I'm taking babies from high-contact group and I'm putting them with the low contact, I'm taking babies from the low contact, I'm putting them with the high contact, that you become what your adopted parents are, your foster parents, and not what your biological parents are. So the amount of your subsequent behavior, including the contact that you're going to have with your offspring, as well as differences in social behaviors that they show as juveniles, it tracks with the foster parent and not with the biological parent. So we said, hey, and this is just showing that. So we said, are there differences in the magnification of this perioral structure where most of the contact is taking place? And this is in progress. We only have a few cases. But it looks like if you map primary somatosensory cortex and listen to what neurons want to respond to, that in high contact individuals, this portion is much larger in the high contact animal than the low contact, and that's not surprising. You're being exposed to much more tactile stimulation very early in development. And this is work that Adele just submitted. And these are really, really hard experiments. So what if I look at the connectivity of primary somatosensory area, especially in this region where they're getting a lot of contact? And what she had to do was in both high and low contact voles, she had to match her injection sites for the size and location, really, really important and very hard to do. She did quite a few animals, and we have only a subset of which were matched. These are injection sites. And then she plotted all the cells in cortex, looking at connections, and was able to calculate densities and connectivity. And this is an example of what a reconstruction looks like. This is rostrals to the right and medials to the top. And this is a low contact and high contact. And you can see, generally the patterns of connections look quite similar. But if you really start looking here, you're saying, that's quite a bit different. And that's quite a bit different. And if I look at the internal organization, that looks quite different as well. So and callosal connections, so connections with the other hemisphere, are radically different. So this is a high contact individual with only having connections with S1 in the opposite hemisphere and motor cortex. And this is a low contact animal. And you can see that the connection patterns are really very different. And she quantified these. And low contact is here and high contact is here. And this is just percentage of percent difference in different cortical fields. So for example, high contact individuals have about 13% more intrinsic connections in S1 than low contact. Low contact animals have about 10% more connections with motor cortex, and so on and so forth. If you look at contralateral label, and she also did some, the groups, the different cases in the groups, you have, significantly more contralateral labeled neurons than in low contact than high contact. And you also have just different areas that are projecting to S1 from the opposite hemisphere. So you have these, this is a pretty subtle manipulation. It's not a manipulation, it's just natural variation within a population. And what you find is that you have functional changes in the neocortex that reflect those differences in rearing styles, and you have quantifiable changes in cortical connection patterns, and she's working on thalamocortical connections, and those are different as well. I'm not suggesting they're completely different, but there are clear measurable differences in their behavior. So I'm going to end with that. This is a nice short talk. What factors contribute to the cortical phenotype? And I didn't talk about genes, and other people will talk about genes. As I said, they can contribute to differences in cortical sheet size, cortical field size, cortical connectivity. Zoltan will talk about that. Genes, I showed you, can generate changes in peripheral morphology and cellular mechanisms involved in plasticity. But the environment can change the internal organization of a cortical field. It can change sensory domain allocation. It can change cortical field size, cortical connectivity. And the environment can actually change peripheral morphology in terms of bone density, depending on what sorts of fruits and nuts you eat, or the hardness of your food when you're developing. I've given you an example of the bill of the platypus and how alterations or radical alterations in peripheral morphology and use can generate enormous changes in the cortical phenotype, and said that humans follow this, similar principles of construction. But things like social learning, language, and culture, I think we can consider these. They're just complex patterns of physical stimuli that vary during development and throughout evolution, and that are going to change can radically change the structure and function of our brain. Not just a little tiny bit and it's going to tweak a little bit here. Or, these can radically change the connectivity. And this is just a review that Danielle Stolzenberg and I did recently. And it's kind of busy, but I'll tell you what the main point is. I call this morphology, but it also includes changes in genes, but physical changes to the human body over time, and environmental social contextual changes that have occurred. So if you look about 6 million years ago, you have the emergence of a FOXP2 gene. Hominids have split from chimpanzees. About 3.2 million years ago, you had features of the modern hand, not necessarily the modern hand. About 1.6 million years ago, you had changes to your supralaryngeal structures. 700,000 years ago, you had a modern hyoid. You had a modern hand, muscular changes to morphology. So you had the physical characteristics necessary for complex tool use and for language, anatomical changes in the capacity of the cochlea and changes for frequencies associated with speech production. FOXP2 undergoes different sorts of changes. What's really interesting, 195,000 years ago, earliest anatomical modern humans. So we had all the stuff. If you look at what's happening culturally, it's contentious, and Gary could maybe talk about this, of when modern language emerged. But it certainly didn't emerge with the emergence of FOXP2. If you look at tool use, it was just large cutting tools, not the fine sorts of tools that we use today. The control of fire comes on here. And if you look, you start getting stuff really rocking and rolling about 300,000 years ago, such as cultural traditions, grammatical language. 100,000 years ago, linguistic diversity. 70 to 50,000 years ago, sophisticated blades, needles, clothing, increased population. Like 125 years ago, or 200 or so years ago, the Industrial Revolution, and only within the last 125 years or so, you know, automobiles, nuclear power, electronic social networking, the use of the computer. My point is that this is happening, you know, it was in place 195,000 years ago, but all of these complex social behaviors and human behaviors came on the scene much, much later. So there must be something other than the structure or the genes that are generating these. There has to be some epigenetic mechanisms that then perpetuate these complex behaviors across generations that has nothing to do with traditional evolution. And so I'm going to end the talk there. I just want to introduce my laboratory and some of the people who are involved in some of the research. This is James Dooley. He's a student here. He did a lot of the monodelphis work. Deepa Ramamurthy is here as well, and she's working on monodelphis, and hopefully she's going to talk to Tony about some of her stuff in the somatosensory system. Adele Selke did the work on voles, and these are other members of my laboratory and people who pay for us to do the research. So thank you. Good?
So that was great. So we have time for some questions, and we'll start with questions from the back, please. And then, so, yeah.
One question, were there any behavioral changes when you think these animals that were born touched by their parents, apart from the fact that they were actually touching more their offspring? Because you see so many changes. So were there
Other behavioral changes? Yeah, and you know, if you're, I can't give you all the details, but Karen Bales, they actually have some publications on this. So they have changes in the social behavior, when they come in contact with a new individual, how they behave, the amount of time they'll spend exploring. So it's more than just the type of parents they become. It absolutely positively impacts a lot of other behaviors later in life, these early tactile experiences. But her last name is Bales. It's Karen Bales, so if you're interested, you should look that up because she has the details on those.
Are both parents generally low contact and high contact, or are they mixed?
They're generally both low contact and high contact. The mother is definitely more high contact than the father, but they're both generally grouped as high contact or low contact. And what we do is we don't look at the middle group. We take the extremes so we can see an effect easier.
What's the time scale of these changes for an individual, for example? I mean, how much time you see like in this rewiring?
Oh, in the voles?
Yeah.
Well, we do the experiments, we do the anatomical experiments when they're about 6 or 7 months old, when they're adults. But in terms of the time scale and when they're occurring, we haven't looked at that. Is that what you mean?
Yeah, exactly.
We haven't done a developmental study. And the next on the list is, okay, what exactly is happening and at what point in development are these changes occurring? Yeah.
Also to understand if there's like any like factor that is really crucial for this time scale.
Right. So I should say the other parts of the study that we're going to do, a graduate student of mine, an old graduate student of mine who's now a professor at UC Riverside, she's going to look at alterations in gene expression to see if, in fact, these differences in tactile contact will change the patterns of expression in genes that are supposed to be involved in axon guidance, for example, from thalamus to the cortex. Danielle's going to look at epigenetic factors in the cortex to see if those are different in high contact versus low contact. So we are pursuing, we just wrote a grant actually, to look at the kinds of things you're talking about.
So, is this working? So we want to use microphone for questions because we're recording the question and answers. So, so don't ask your question until I come to you with the microphone. So Gary was waiting to ask a question.
If you go back to the last slide before this one, I have two comments. The first is the situation is probably even more extreme than you suggested. So I don't know what you're thinking from modern language 700,000 years ago, but I would actually put language more recent. I mean, I've seen different estimates.
But that's why I said it's controversial. And
yeah.
I have to say, Gary, Danielle and I, this is in a recent review we just did, and this comes from a lot of different sources because it's a little bit out of my area of expertise. But the language thing, it's really controversial because some are
yeah.
saying 700,000, but then some are saying, no, it's more like 300,000, and some are saying it's 40,000.
You need like bracketing bars.
Right.
But
I mean-
anyway, so I think, if anything, your point is even stronger than you've drawn. And I would put the cultural traditions a bit more recently too, but again, depends on what source and how you define it.
Exactly.
And so forth. But
Because that was a hard figure to make.
if anything, the general point that you want to make is even stronger, which is there's all this stuff that's very, very recent. But the second part of what I want to say is I would flip the question around slightly from the way that you framed it, which would, the way that I think about it is there, for example, changes in intrinsic connectivity that led to the culture stuff? So it's clear that what's driving a lot of this is culture, it's language, it's being able to communicate how to do things, technology, and so forth. And the question is whether there's any intrinsic, maybe genetic change that allows some kind of circuitry that allowed this sort of branching off. And we really don't know the
We
answer.
don't, but I agree. I don't think I would take such a strong stance that it's all culture, that there's nothing happening even. There is, and what's coming first? What was the paper I read? It was the cultural capacity. There are lots of papers now on cultural capacity, and are there these intrinsic changes in the brain that then allow for this kind of stuff? But then where do the intrinsic changes come from, and how were they generated?
So to go back to that slide towards the beginning with the A through G, where I wanted H.
Right.
With the sort of 7 mechanisms of change or something like that. My own gut instinct, and I don't have the data to show this, but coming from an understanding of cognitive psychology and an understanding of computation, maybe an idiosyncratic understanding of computation, is that we probably are looking for some kind of circuitry change. The thing that I think about is the difference between a computer program that has access to a bunch of memory locations and one that has access to what they call in the language Lisp, a linked list, where you can have an organized structure of memory. And to me, maybe that's what's the fundamental thing that takes you, let's say, from the pre-change to the post-change, or from the Neanderthal line to the human line, is maybe there's a way of organizing your memory such that you can have structured representations of things. And that might be, if that theory were right, that might be an intrinsic connection. That's part of why I pushed earlier, is I wonder if we'd ever find the answer with A through G for what's changed. Maybe what we're really looking for is some kind of very fine-grained microcircuitry change that changes the structure of memory.
I agree. I mean, that's why you weren't here for the intro to the intro, the first part. It's like we can't, I'm just looking at a systems level, but absolutely, and now I'm pushing, which, if I'm interested in the brain, I have to also look at a social level, et cetera. But you also have to look at macrocircuits and microcircuits and neurons. I mean, and those things are changing as well and impacting these higher levels of organization. And how that's occurring is really tough. But we have to cross these multiple levels to try to figure that out. And just because we know about some lower level, it doesn't necessarily mean that we know how it emergent properties of neurons, form coherent thoughts, for example. But there's a tendency to want to, that's why we're all gene-centric now. There's a tendency to want to break it down to this, like, okay, now we know about genes, it must all be genes, or we know about individual neurons, and it's just about all about individual neurons. But even by recording from neurons in an awake behaving animal, we still don't know how the brain works. I mean, I'm rambling right now, but I get what you're saying. Okay, Tone?
Okay.
One word to that.
One last word.
The last word is circuits, is where I...
Yeah.
So, more questions from students? Yeah. So we're going to have, I think, another 5 minutes of questions, and discussion can continue after that at lunch. But also, we're going to use the Twitter hashtag #BCBT2014, so you can put questions there. So who was it? Had a question?
Don't do questions for me on Twitter. I don't do Twitter or Facebook. I know, I'm a little behind it. No, you have to see my cell phone. He's always saying, "Oh, geez, we got to get you a smartphone." I don't want a phone that's smarter than me.
She stole it from a month ago.
Okay, so I have a question which is slightly related to the time aspect. So what I want to know is, have you seen, or do you know if this switching parents experiment that you did, if they exhibit some kind of critical period with respect to this behavior, beyond which they cannot learn the other kind of...
We haven't done that, but that's a great question. I mean, that's also important. So if you swapped them out, like after P4, these are swapped at the day of birth, right? So if you swapped them at P2 versus P3 versus P4, I would suspect there's a critical period where you're not going to see the sorts of changes that we're showing. But that's a really good question.
question here?
So it seems that from the behavioral level, you can see changes in connectivity and changes in density of areas. The changes in connectivity, I can think they may be explained through the Abian rule by the fact that two areas are more activated together. But the changes in density, is there any clue that would suggest that an active area attract more neurogenesis somehow?
I don't know. Or a decrease in naturally occurring cell death. So I should say with Monodelphis, well, at least with Monodelphis domestica, neurogenesis is occurring until P18, but in rodents, neurogenesis stops postnatally, correct? So it couldn't be an increase in neurogenesis. It would have to be a decrease in naturally occurring cell death to account for the greater density in neurons we see in, let's say, wild-caught rats versus laboratory rats.
Yeah, a lot of possibilities because there's been years of selection as well, generations and generations of selection.
Right.
Okay, thanks.
Any more questions?
Let my people go.
Now, look, so you, on the previous question you said, oh, wait, I'm going to show you the ultimate experiment that makes everything clear, right? And that was the rearing, the parenting experiment. Okay? So
why do you always have to rain on my parade?
no, wait, so this will be fun, don't worry. So, so there are two things here. On the one hand, so how one of the principles that we have identified using robot work is something that we call behavioral feedback. Relates a bit to Gary's point, where you don't have to say, oh, I have to develop some magical circuit property to do something. But what we show is that if you structure your environment, it will automatically start to structure your behavior. So if you want, you start to have habits that are also fully externalized in how you structure your environment. But as soon as you start to have a habit, the way you sample your environment will start to systematically change.
And it should.
Right? Exactly, and this will have an
That's
immediate
adaptive.
impact.
That's an adaptive thing to do.
This will have an immediate impact on your perceptual and cognitive systems, as you also show, right? Because you show, look, if I change properties of the environment, I start to see changes in how this nervous system is organized and classifies the world. But now we want to see this link to behavior, and that was your parenting experiment, right? And there, I could challenge you and say, well, this is cool. However, maybe what you're showing to us is more a non-specific effect of, let's say, enriching an environment, as opposed to really the circuit that underlies the behavioral change, right? So how can you convince me that this is really specific to the behavioral change in parenting, as opposed to just a non-specific enrichment effect?
Okay, so here's how I would be able to do that, and hopefully this is what we're going to do, is that in these animals, we just took high and low contact. I think what you want to do is cross-foster, and then so you cross-foster half a litter. And then you compare, and this litter is raised with its biological parents, and this litter is raised with the cross-fostered parents. And then you start looking at connectivity and receptive field properties and say, okay, so if what I'm saying is true, because right now I just showed you a correlation. I agree, right? If what I'm saying is true, and then you also look at gene expression in both, you look at epigenetic markers in both, for example. So, and then you compare the biological litter with its cross-fostered litter and say, okay, does this look more like the parents, the biological parents, or does it look more like the cross-fostered parents? And what does this look like? And I think that would be your answer. They should both be different, first of all, and they should track with what they were raised with.
Another question?
No. I wasn't. Yes, I buy that. But there was another element that maybe I wasn't clear about. Like in your anatomical analysis, you looked at, for instance, the sensory to M1 projections. But now you could argue, well, but these parenting behaviors, those very much stereotyped behaviors, they're expressed by brainstem structures, or very primitive, like the central gray, for instance. So if there are any kinds of changes, it is how, let's say, cortical areas are controlling subcortical regions. So is really a pure cortical change sufficient to really tell me anything about these kinds of changes in behavior?
As far as I'm concerned, it's just the tip of the iceberg. We're looking at thalamocortical changes. You want to look at cortical are projections from the cortex to the brainstem, the colliculus, to the developing thalamus. And so the point is if you wouldn't even have to have enormous changes at any of those structures, but if you had slight modifications based on how you're reared, the degrees of freedom for processing is enormous. Yeah. So this is just the tip of the iceberg, and it is. I agree, it's correlative, but I think there are ways that you can really, nail it.
So I think we should move on, but John, you need to set up. Yeah. So while John's setting up, I'll ask you a question. Go on, one more. In your comparison between the wild and the lab rats, so you're showing these differences in S1, also overall brain size, is that right?
Not overall brain size.
Okay, but S1 and M1.
Differences in auditory cortex and somatosensory cortex in terms of size, and then we looked at the density of neurons in P1.
Yeah. So my question is, so you're probably going to want to assume that most of these changes are occurring postnatally, or because you're saying this is
that's what that's what I would think.
And probably then in the first 2 weeks postnatally, because that would be when the cortex was maturing.
Yeah. Can I just cut you off right here and just say one thing? So Dylan Cook, who is at UC Davis in the Department of Psychology, he and Danielle are actually doing really cool experiments where they're rearing laboratory rats out in field pens out in the wild to get at the, sort of nail that question down too.
Okay. Yeah.
And looking at changes in the cortex.
Yeah, so that was really where I was going. Can you rule out strain differences between?
Well, no. So that's exactly right. There could be some strain differences, but they are rearing, and you should see the videos of the behavior of these laboratory rats that are put, and it's hilarious. So I don't know what you guys are like with your animal research, but we have our people who come and they said, the animal room has to be between 67 and 70 degrees. We take the laboratory rats, they give birth out in the field pens. Well, they do, I don't. The first day they went out there was 104 degrees, and then they're living out there at 30 degrees in the winter. So in the videos of these animals are extraordinary. They're behaving very differently than they do, and you could talk about that. Oh, good, yeah. So there are ways to get at these things. I mean, this is just, like I said, we're just, this is the tip of the iceberg.
Okay, well, thank you very much. It's a fantastic kickoff talk.